So, welcome to all of you all once again. I'm really extremely delighted that we're going to hear our interns presentation today. We had 10 interns in the US and distributed between UMD and Nebraska. We will have, we also have an international student who's working with Stevany. She did a virtual internship this year. So let's get started. I don't want to waste any time. I welcome the first presenter, Rosie. Go ahead.
Hi everyone, this is Rosie. I'm an incoming undergrad senior at UCLA, and this is my project that I did over the summer. So I had the honor of working under Dr. Liang and Shun Chao to research about the key climate features driving corn yield across the Corn Belt. Yeah, for an intro of my project, as you can see, this is an image of the Corn Belt states. So they're basically the regions core to the U.S. corn production. And as one may expect, corn yield is highly affected by climate variability. Previous research has used climate variables to create models to predict corn yield, but they tended to use a lot of fixed variables and seasonal definitions, which had limited interpretability. So because of that, it may become difficult to find the significant driving forces behind the yield. And in my internship project specifically, I wanted to tackle this question, and hence my research question, which is that what predictors have the greatest impact on corn yield? Okay, so let's jump into how I created the data. So basically, I tried to merge together the USDA yield data and the Met basically daily weather data consisting of temperature max, temperature min, precipitation, and snow. So basically, well, these are just more detailed steps of how I merge the two. But basically, what I did is that I extracted a few columns from each data set and I merged them together using county name, year, and state. So that way I have basically annual county data um, of basically the metrics that I wanted to explore, as well as the yield attached to the right of that. So with that setup, I could manipulate the data a lot easier. So now, the first model I tried was just a simple random forest with like um, basically no tuning variables and no feature um, engineering. And basically, yeah, the accuracy is not too high with like barely a 0.3. And with linear regression and other attempts, like I basically just didn't try to go further because they were far too low accuracies. So given this baseline model, I wanted to try to improve the accuracy and identify basically what are the most important features that was driving corn yield. So by trying to find what those features are, I hypothesized I would be able to improve my accuracy. So first off, um, I was thinking to try to cluster the data into a few different groups because as you guys may know, like data forms into like natural clusters, um, like very naturally. So I wanted to take a look at like what are the ideal number of clusters to use in terms of data points with least variability across the regions. So as you can see, I ran like a k-means model and I was able to find these three clusters. Um, and as you can see, like in this plot, like I chose three because the within cluster kind of variability had decreased um, has decreased like dramatically and it kind of tapered off. So this is kind of like the ideal point that I try to use in order to like minimize like computational efficiency as well as like the variability within the clusters. Okay, so here is where I try to engineer different features based off of what I had already been given. So instead of just having like T max, T min, um, and like precipitation snow kind of like annual means, I tried to divide them up into different seasons throughout the year. And at first I had like set these very like default numbers um, down, as you can see, like two-month periods. And I also tried to make like seasonal, not just seasonal means, but anomaly features, such as like what percentiles kind of like for all of these different features could maybe like if I were to add these features into my model could make this model's accuracy a lot higher because of these like anomaly events that have occurred. And I exploit those throughout these all four of these different features that I was given. And yeah, at first like I set these default numbers as I listed. And this was in attempt to improve accuracy of my model. So of course we can't just set like these default numbers forever and expect them to do really well. So I tried to basically use different algorithms in order to find the most basically the thresholds that would allow for the highest accuracies. And yeah, in terms of these like seasonal ranges, I also um, did a sort of like search algorithm on that. So yeah, for each season, I set them to be like at least 14 days. Well, I call them seasons, but it's mostly like kind of periods that are most significant to kind of like tell you what is like the significance like behind the variabilities, basically, if that makes sense. So here are the percentiles and here are the ranges that I found, as well as like the clear day thresholds. So this is at first I tried using the binary search algorithm, which is a lot um a lot less computational expensive, but um it wasn't as like effective because there was like it was mostly like a zigzag pattern throughout the years for detrended yield. And so that's why I tried to use a random search algorithm, which we just used. I tried 200 iterations, but I also tried it again in order to see if I could get even better optimal percentiles. So yeah, this is how I tuned the hyperparameters basically. And then, so after tuning the random forest model, I was like, okay, how about let's try to build like more models or more sophisticated models in order to see if that could additionally improve accuracy. So I tried the stacked model, which is random forest to gradient boosting and then to MLP. And these are just basically different like layers of models that you put on top of each other in order to like increase accuracy. And then residual stacking, basically I try to correct bias by using like residual. That's kind of like how it works, basically. And then oh yeah, and then I tuned a bit more using like PCA features, which means like I found the features that have the most variability throughout like the entire model, and I picked the top two, and then I filtered the NA values not by dropping rows, but by using like a more sophisticated mice method in order to like prevent like bias because of missing data. And then I tried removing bias another way by adding like a layer of linear regression. So here are kind of the results, yeah, of like the all the models that I tried talking about. And as you can see in the very end is where or the very last column is where I got like the best results. It wasn't necessarily from the more sophisticated models. It was more of how I try to clean the data, if that makes sense. So I basically used a simple random forest model, but I tuned the data using the random forest model, and then I tried to take care of, like, for instance, like NA values a certain way, and then removing bias. I found that to be more effective than just like, I don't know, stacking models on top of each other, making more sophisticated ones. So yeah, this is for like our best model. And then I basically did a plot for like the annual correlation coefficient. And as you can see here, like the clusters are very, very different. Even though like the second and the third one seems like they're mostly static. But as you can see, like the reason why these clusters are made is still because like these clusters are still very much different from one another. Um, and And yeah, so you can see that cluster, the leftmost cluster has a maybe like a slight increase, even yeah, even though like the other two stayed mostly the same, but they're still very much different, as you can see from the graphs. Um, yeah, and then I took a look at the most significant features by using like feature importance graphs, and they mostly stayed s- stayed the same in terms of the very most important features. But as you can see, like the engineered features that I put in has had a really great impact, such as the PCA, the anomaly, Tmax. So those definitely did um, like we can see this is why the models where I feature engineered and tuned was a lot better than previously without like any of those basically manipulations. Yeah, and then so that basically concludes my results, and then so now for limitations of my model, I want to say that the biggest thing I realized towards the end was that because I had tuned my my model using my random forest model, so like basically I tried to find w which are the thresholds and the numbers of my featured and my engineered features that yielded the highest accuracy for my random forest model. So basically I tuned using my random forest model, which is probably a big factor of why like those stacked models didn't work as well using the tuned parameters that was tuned under the random forest model. So basically, I was thinking that in the future, to continue this project, I could try to tune the parameters using different models, and then perhaps that would yield better results in these specific different models. Um, and yeah, so the other thing is computational and memory limitations of just like working with code in general. And I tried extracting other variables such as like the solar radiation, and it wasn't working. But I think I believe that more computational efficiency in the code somehow could potentially solve that problem. So that's another step that I was thinking of taking later on. Yeah, and then I would really like to acknowledge like everyone and just I don't know, everyone who's been like a part of this process, especially my mentors, like Dr. Xinzhong Liang and Chao Shun Chao for their like unwavering support, and they're super busy, but they still made a lot of time for this project and was super patient. And a lot of tech support came up too, and yeah, they were just really, really genuinely helpful. So I sincerely appreciate them for that. Um, and yeah, like my where I had gotten the data from USDA, the Prism Climate Group, and then I used a server from Zartan. So like I could not have done this without like all of these resources and people. and funding support from the Dawn internship, as well as my ASIC colleagues for basically guiding my project and then for like navigating this new like living environment at UMD because I'm from LA, so I knew nothing about the East Coast before, but they were also super helpful for that. So yeah, that's it. Thank you so much for listening to our presentation.
Hey everyone, my name is Vincent Ferrera, and I was also part of the Dawn interns uh, here at the University of Maryland, along with Rosie. And my research project ended up being just evaluating the linear regression performance on historical U.S. Corn Belt corn yields. Now, originally, I wanted to assess the role of extreme precipitation uh, in damaging U.S. Corn Belt crops via flooding or drought. But unfortunately, that research goal was adjusted due to time constraints. And one of the kind of reasons why was, one of the first things I needed to do was to spatially link the detrended county crop yields and the meteorological data. Now, I know Rosie talked about how she was able to do that with her research, but I was unable to do so. So my focus kind of shifted to just solidifying the analysis of the spatiotemporal variation of detrended county corn yields in that Corn Belt region. Now, there is a well-established positive trend when it comes to U.S. crop yield time series. There's been a lot of research papers in the past identifying how improved management practices, technological advancements, crop genetics, and other factors have just contributed to yields increasing over the years. But there's been several yield detrending strategies that have been used in that previous research in all these like papers that we've looked at. So, for example, like some have used linear regression, some have used decadal averaging or multi-year averaging. Some used running means, like a several-year running mean, to detrend it. Others have used coefficients of variations. Now, I guess what I wanted to ask was, if we took, say, a simple linear regression approach, where can this be appropriately used to detrend, say, corn yields, and how well can it do so? So here's just kind of like the methods that I took about to answering that question. So data I just obtained, or Dr. Sun was able to provide me with U.S. Corn Belt county level corn grain yield for the following years from the USDA Quick Stats data page. The coding environment I used was in Python via Anaconda and the Jupyter notebooks, and I used PyGIS packages to kind of help me out with that. And in terms of my analysis, I just kind of made several combinations of just kind of the following variables. So I took all of the U.S. Corn Belt states kind of one by one, selected an administrative division, whether it was county or district, selected the value that I wanted. So if I was looking for just raw yield values in bushels per acre or a ordinary least squares regression, detrended yield, also in bushels per acre. And then the aggregation technique, I've made either a kind of line plot time series of these yields, or I made a state map that kind of plotted the year by year kind of yield analysis for each of these states. And in terms of evaluating the performance of this ordinary least squares regression, I did it kind of two ways. First, I looked at the R squared values to kind of see the the explained variance in the yields with time. And I also looked at the p-values for each of these performances to see in which states are they statistically significant or not. And all of these were aggregated at the state level across counties or districts, depending on the plot I used. And the mean, minimum, and maximum of these values were obtained. And I also provided a plot of just the linearly detrended corn yields And I provided statistics at the top of those graphs of the state means of the average, standard deviation, minimum, and maximum detrended corn yields across the counties and districts. So here's just kind of the first results that I have. This is an example of a good example of how the ordinary least squares regression, had a good performance in, say, Indiana. This is the average Indiana county corn grain yield. And we can see that obvious linear trend with time from the 1950s all the way through the mid 2020s. And when we take the detrended yield, we can see that it's mostly a straight line. There are a few outlying points here where we see some severe yield losses, and I'll touch upon that when I talk about the spatial plot. But we notice that the R-squared values are relatively high, explains 83% of the variance, across the states for Indiana, and the p-values are basically zero for all these. So, here's an example of a good usage of ordinary least squares for a state like Indiana. In contrast, here is a state, Kansas. Kansas did not perform, or I guess I should say the ordinary least squares regression did not perform well for states like Kansas. And, we can see that the spread among the counties is a lot greater than what it was for Indiana. And the linear trend is not as obvious. R-squared values drop to under 50%. And the p-values for some of these counties are much greater than 0.05. So, for states like Kansas, I wouldn't trust the ordinary least squares regression for detrending purposes. And we can kind of see that along the right side with the detrended plot of the huge spread between the counties and the kind of back and forth, up and down nature where it didn't really exactly detrend it completely. So here's just kind of a state-by-state regression performance summary that I made. So you can see that states kind of, east of, this kind of, east of these kind of Great Plains states here, that all of the p-values, even the max p-values are very, very low. So the ordinary least squares regression can be considered trustworthy for, all these states here, but not for Kansas, Nebraska, or South Dakota, where the p-values can get very high. Now, here is a state plot, or kind of the spatial variability of the yields. So down here, we can see the detrended yields, so normalized. So blue is like positive yield anomalies and red are negative yield anomalies. And we can see, or we should be able to see as we get to certain years, that some counties or some entire regions or the entire state just get really dark red. So, this is a great way to, kind of visually see, years or locations where severe yield, like severe negative yield anomalies can be seen either in certain particular locations or across the entire state, like just now. I'll just kind of leave that on for a minute. How much time do I have left?
You have another 1 minute.
Okay, I'll move on then. So in terms of just discussion, in terms of interpretation, so like I said before, the ordinary least squares can significantly detrend corn yields in most of the states, except those kind of Great Plains states. And it explains around 78 to 85% of the corn yield variance with time in most of those statistically significant Corn Belt states. And that spatial time series plot, effectively located, county years with severe negative detrended anomalies. But we can see that those events vary in scale. So there's different kind of causal factors for each of those events per year. In terms of limitations, the ordinary least squares, regression does not predict future yields by any means. So, I wanted to allude to a graph taken from, one source where they implemented climate models to predict future yield impacts. And we see that all those models predict like negative yield trends in the future. So, if we apply the ordinary least squares to the future, it would predict positive yields in the futures, and that directly contradicts what these kind of climate models are predicting. So just wanted to keep that in mind. It also may not be the best detrending method. So like I said, kind of at the earlier part of the intro, there are many other detrending methods. So there could be better performing ones, even for those states where it did perform well. And the results might vary in other, countries, other US regions, or for any other crops like wheat or soybean, which are also really important in the Corn Belt region. And there are other evaluators for crop performance as well, besides yield. So crop production in place of yield can also be used, or if you want a real-time evaluator for crop performance or crop health, you can use something like leaf area index. So those are other options as well. And in terms of a potential future implementation, kind of connecting back to Rosie's research, these OLS results can serve as validation and or caution to usage of such linear regression detrending on more advanced data like those machine learning algorithms and random forest things. And eventually, I would want to kind of apply these to meteorological data, link the nearest stations to those county district points for future correlative analysis. And here I've just kind of relisted those conclusions here. I can leave it on this slide, but I do want to just quickly acknowledge all of these people here for advising me, providing me some code, providing me some general advice, and for funding and supporting my internship here. So thank you all. I wouldn't be here without you, and all of this wouldn't be here without all of your support. So thank you very much.
So hello everyone, my name is Tokhirjon, so I go with John as well, Tokhirjon Vokhidov, and I had an honor with working with Dr. Gusharma Balboa. I'm also from Dawn, and yeah, let's move to the introduction. So I born and raised in Uzbekistan, which is located in Central Asia, and I'm studying computer science at the University of Arizona, and I'm on my senior year, considering to continue with grad school. And I did this internship remotely. Yeah. So the project focused on, the project name is creating a searchable on-farm research with Python and GPT. So the project focused on digitizing and analyzing 10 years of Nebraska on-farm research, nitrogen trial data, and it's specifically focused on 2014 to 2024. Yep, and I used GPT to help answers like complex research questions, going beyond basic filters to explore patterns in yield, nitrogen rates, and p-values, tillage, and country level trends as well. County level trends, yeah. And then alongside with that, I also wrote Python scripts to extract and structure key data from raw PDF and image-based trial reports, which allowed for more efficient and standardized analysis. So it's the introduction of on-farm research network, which is also OFRN. The on-farm research network is a collaborative program that works directly with Nebraska farmers to test agronomic practices under real field conditions, and it's a great example of research done with and for producers. And also each year they publish a comprehensive research book. Like here we can see the front page and back page of this 2024 OFR book. So it also contains over 150 pages each book with detailed studies here. And the cover you see in the middle is like showing the county level distribution across the state, including the different topics and different studies here, as you can see. And on the left here, you can see the example of a single study page. Each study contains detailed information like crop type, irrigation, treatment rates. Here we can see treatment rates and the yield as well. And then but it's not in the form that's easy to search or analyze. So we decided to move to the database. Yeah. Here is the database. Yeah, this is the database that I talked. This database contains like over 1,000 rows of curated trial data from the 2017 to 2024. Yep. And then the most important columns here is covers, crop type, and tillage, treatment yield, p-values, and so on. So to handle this scale, I wrote Python scripts. Here you can see some part of the code using tools like PDF Plumber, PyTorch, and Camelot to extract tables and metadata from both text-based and also scanned PDFs. You can see a snippet of the parsing code here. Yeah. And also the output from this scripts was compiled to a single CSV for easier querying. And then I built a GPT-based AI interface shown here that can answer natural language questions using just the structured data. It goes beyond simple filters and supports more complex queries like average yield trends, most rotated crops, or tillage patterns as well. Yep. And then we move on to project objectives. Yeah. So the first is to digitize and standardize over a decade of Nebraska on-farm research data into one clean database. And the next one is automate the extraction process using Python instead of manually copying data from hundreds of pages. And then finally, to build an AI tool that enables interactive insights, also making it easier to explore trends and answer more complex analysis and agronomic questions as well. The methods we used is first one is data collection. I collect over a decade of Nebraska on-farm research reports, and then I extract trial level information using OCR and also a table parsers. And the next one is I cleaned and structured the data into a consistent analysis ready format. And the last one is I integrate it with AI to allow natural language querying across the entire dataset. So the first one is the database overview. Here's the results we see. On the left, you can see a screenshot of the actual spreadsheet we created. Each row represents one agronomic study here, and we can see here we have our 1,000th row, which means our 1,000 studies. And also, I focused only to 2014 to 2024 because we liked some information and some inconsistencies on 2017 and yeah, on 2013. And also, there are over 115 columns, which captures a wide range of information like year, crop type, and so on. On the right here, I told about like a structured since each study took like 4 to 5 minutes, so overall it takes like over 80 hours and so on. And also for the cleaning, it took like around 30 hours as well. So what it gave me a structured analysis ready format, and that enables advanced filtering, trend tracking, and also AI-powered querying. Of course, there were some challenges, and one was format that enables one was the inconsistencies that I told about earlier, and these study formats and terminology varied year to year. And with so many columns, simplifying the data for visualization was something tricky. But overall, the structured format became the core foundation for all of the AI and analysis work that followed. And then data extraction using the Python, yeah. So at the top left, you can see the snippet of my code. And here you can see the result being here. It takes like up to like around 4 to 5 seconds for one study to process and to make it into a CSV format. And here we can see the CSV format that we got through this study. Yeah, here is the title, ID, and yield as well. And here we also lacked some information because of the inconsistency. We filled this with an A, and so yeah. So like raw output sample, like title I showed here, and some like the thing is, we could also upload the PDF and also upload the JPG, but we prefer PDF because it's more like clear to this kind of code. But we couldn't find a general code since some inconsistency, and we could success like a little percentage and not so much. But when it comes to the benefits, this automation reduces time, like processing time, like 10 or 20 times than the manual one. And it worked on both scan PDFs and the JPG as well. The challenge I had, like most of the sites like length of the treatment's name table, and there are like more than one table having two or three, and these tables like most of them didn't like relate to the study as well. And OCR also return some kind of bad errors and like noisy clouds as well, and so on. Yeah. And the third one we got is Nebraska on Farm Research GPT tool. It's an AI tool. So at the top left, you can see the chat that we created with GPT. So it analyzes our 1,000 studies, and it also answers in more natural language questions like. So it's a better version of Result Finder. If we go to the Result Finder, this one, yeah, it's here. In the Result Finder, we can see it's just one filter stuff. We can like choose county and year and crop if we want and so on. And we can just tap search, and we could get the studies here and so on. But with this one, yeah, with this kind of GPT, let me start new chat. Yeah. So if we ask about this, like about the counties with most frequent one, here we also have a database. If we also have to filter it from 2014 to 2024, and with county here, just showing sound cycle. Yeah, apply sounders. Or we can just go to this one, and we can see this like for nitrogen one, we see this in the sentinel nitrogen, we got a rate of 181, while a grower had 193. And for the yield wise, it did analyze in two methods, like by treatment wise and by study wise. In both of them, sentinel got like 7-ish bush per acre more. This means that sentinel gave better yields using less nitrogen, which means it was more efficient and could save money while protecting the environment as well. Yeah, this kind of simple questions can be answered with using this GPT tool. And also like the key benefits is quick, intuitive, and also can handle multitask variable questions, making it ideal for demos, outreach, or exploration as well. So one like limitation is that the dataset needs to be updated as new reports are published. And so to wrap up, we can say this, like, first, we found the structured data is essential for deeper, more reliable analysis, especially when used with AI tools like GPT, which thrive on clean inputs. Second, while we built automation tools, only around like 10 to 15% of studies could be successfully extracted using code due to inconsistent formatting and lack of structure across the original files. And like lastly, GPT Assistant allows users to ask natural language questions and also get fast summaries or comparisons across hundreds of trials, something that would take like maybe hours to if we do manually. And the lessons learned is brief theory with practice. I applied my theory here in internship and also built practical skills as well. Since I worked remotely, it gave me this discipline to work here in internship, and also we could show AI in action while customizing GPT on our own, and also gained research insight here. And yes, thank you for your attention, and especially to Lena uh, for helping me through the process in this internship, and to my mentor, Professor Dr. Guillermo Balboa, and to you, everyone. Thank you.
Okay, hello everyone. My name is Daniel, and today I'm going to be talking about the project that I did over the summer, which is irrigation water treatment effects on leafy greens. So the table of contents goes as follows: the introduction, objectives, methods, the results, conclusions, and I'm going to talk about additional work that I did other than my project over the summer. So introduction. Over the past couple of years, there's been an increased demand for sustainable methods for growing leafy greens, and this has led to a larger interest in certain alternatives, such as harvested rainwater. And harvested rainwater is good because it reduces the reliance on treated municipal water, and that helps to conserve energy because it takes a lot of energy to filter, um, chemically filter the municipal water as well as pump it. So to better understand the influence of water quality on plant health and nutritional quality, the project explores the different effects of water treatments on leafy greens. So in this project, we analyze certain phenolic compounds. Phenolic compounds are plant secondary metabolites that have antioxidant properties, and they are found abundantly in leafy greens. In plants, they help to protect the plant against stress, such as pests, pathogens, and the environment. And in humans, when we consume these plants, they contribute to our health by reducing oxidative stress as well as lowering the risk for chronic diseases. Their levels therefore reflect the plant physiological status, which makes them a good indicator of the plant health and nutritional quality. And to the right, these are the specific phenolic compounds that we analyzed and their structures. And one common ground between phenolic compounds is the fact that they all have benzene rings in their structures. So the research gap that we aim to fill up with this project is the fact that there's limited data on how water treatments affect phenolic compounds in leafy greens, and the fact that harvested rainwater is quite underused in research despite its sustainability potential. So therefore, this study compares 4 different irrigation sources to assess both safety and nutritional quality. I'll move on to the objectives. So this study aims to test the impact of 4 different irrigation water treatments on Nebraska mix of leafy greens, and quantify and compare the concentrations of phenolic compounds between treatments. Now methods. So this is a study design. The study was conducted at Plantation Park Heights Urban Farm in Baltimore. The different water treatments that we used are as follows: municipal water, which is pretty much just tap water, harvested rainwater, ZVI filtered water, which stands for zero valence iron, and this uses iron in its metal state to remove contaminants by binding and breaking down things like heavy metals and nitrates, and PAA treated water, which is peracetic acid, and this disinfects the water by binding and breaking down things like bacteria and viruses that might be in the water. The phenolic compounds that we chose were chosen for analysis due to their known major antioxidant and health benefits in humans. And by measuring them, it's going to help to assess how the water treatments might affect the plant's nutritional quality. So these are pictures of how the farm was set up. So to the left, you have the greenhouse where when it rains, it runs off the roof into the blue basin right there. And then the blue basin is there because it collects the initial dirt that might be accompanied with the rain. And then as the water rises, it then falls off into the bigger tank to the right to make it which is gonna make it cleaner. And in the picture to the right, you can see this is how the different water treatments were set up. The blue basin to the right is regular rainwater, and then to the left is the peracetic acid, and on the bottom is the ZVI filter. So this is pretty much how the farm layout was. Each bed had the municipal water, ZVI, rainwater, and PAA treated water on one bed. And there was a total of 3 beds, so a total of 12 treatments as a whole. So this is a lab analysis. We collected 2 leaves each from all of the 5 brassica species that were in each treatment bed. And then we lyophilized these samples, which pretty much means we freeze-dried the samples and weighed 10 milligrams of this freeze-dried samples. Then we extracted these phenolic compounds in a solvent made of 70% methanol with 5% formic acid, and then we put it in a 1-hour sonication water bath. We then centrifuged briefly and filtered the extracts. With these extracts, we measured the absorbance spectra using a UV spectrophotometer for the 12 total samples, and we semi-quantified the phenolic compounds based on characteristic absorbance wavelengths. Now to the results. So this is what the absorbance spectra looks like that we took for all 12 samples, and each one of the lines represents a specific treatment in a specific bed. So like MA stands for the municipal water, bed A, bed B and C, and so on and so forth. And on the bottom of the wavelength, you can see that there are specific wavelengths that these phenolic compounds absorb at. To then record the absorbance that helps us to calculate the concentrations. So these are the mean concentrations of the compounds in milligram per gram tissue. The biggest observation we made from this was that the ZVI treated water had the highest concentrations of every single compound, whereas the PAA had the lowest concentrations across every compound. And another observation we made was the fact that the catechine phenolic compound had the highest levels for every treatment. So in conclusion, there are no significant differences in the plant growth between the water treatments, and this indicates that harvested rainwater is just as effective as the other irrigation sources, and it confirms that using rainwater is a viable and sustainable option for irrigation. And the references. And now I'm going to talk about additional work that I did over the summer. So some other tasks I was involved in included sampling leaves, collecting stomatoconductance data, as well as collecting kale biomass data. I also recorded lactic acid bacteria, yeast and mold Petri films, which pretty much means like counted the bacteria in the yeast and mold. Same thing with Pseudomonas bacteria plates on the right. I also prepared microbiological media. I poured some plates, before drop plate method for plating bacteria, and I conducted salmonella inoculations via needleless syringe infiltration of the leaves, and that can be seen in the picture on the right. So I'm going to acknowledge the Dawn CONSERVE Global Future Alliance for giving me the chance to do this internship. The Plantation Park Heights Urban Farm, which is where the project was done, and everyone in my lab, Dr. Mikalev, Claire, Diction, Adam, as well as Farmer Chippy, and the funding source, which is Hari Hughes Center for Agroecology. So yeah, thank you.
Hi, everybody. My name is Sachin. This summer, I had the pleasure of working in the WOW lab under Dr. Goldstein, looking at analyzing water microbiology, microbial water quality in Baltimore. And I'd like to present my experience to you over the next 10 minutes. Before I begin, a little bit about myself. As I said, my name is Sachin. I'm a rising sophomore at the University of Maryland on the pre-medical track. And I am planning on continuing working in the WOW lab for the next semester. So in this presentation, I would like to cover some background on the project I worked on, the methods for analyzing the water quality for the samples we received, the results of our analysis of these water samples, and the impact in connection to public health of our findings. So to begin with some background on the project, I worked on the WET project. WET stands for the Water Emergency Team, and their mission is to evaluate the public health impacts of sanitary sewage overflows and sewage backups in underserved Baltimore neighborhoods and translate these findings into community outreach and policy action. The overarching premise behind a lot of their work is that Baltimore's stormwater infrastructure and sewage infrastructure is very antiquated. And during water events, like when it rains a lot or when it floods, sewage ends up in urban waterways and in people's homes. And so for this reason, the WET project has two main targets. And so these two targets are the environmental side and the household side. The environmental side, which is what I spent most of my time doing, was we would periodically sample different Baltimore waterways that experienced sewage discharges recently, and then analyzed these samples in the lab for bacterial concentrations, pathogen presence, antimicrobial resistance, among other things. And on the household side, you would sample homes that recently experienced water events inside, survey the residents, do the same analysis that we do for the water samples to these home samples, and importantly, translate these findings into an outreach program to these affected communities. Oftentimes, there is the WET team is at church services and local events just to make sure we connect our work to the communities we're trying to help. I mentioned our analysis, how we analyze these water samples, and I will get to the methods on how we do this, but to understand those methods, I think it's important to understand this one key topic, and that topic is fecal indicator bacteria. So what is fecal indicator bacteria? The much of the lab's work and my work this summer rests on the fact that E. coli and Enterococci presence suggests fecal presence in water. This is because E. coli and Enterococci are two bacteria that are present in the intestinal tracts of many mammals, including humans. And so, if they are present in the water, that is indicative of there's a fecal presence in the water as well. Fecal presence in the water is correlated with pathogen presence, things that make you sick, like salmonella and cholera. However, these pathogens are often very difficult to, or at least more difficult to work with in the lab than E. coli and Enterococci. They're often present in very low concentrations, sporadic, hard to grow. So we're able to look at E. coli and Enterococci as an indicator for pathogen presence in the water. So for this reason, the EPA sets standards for fecal indicator concentrations in a wide variety of water. But in our case, we're looking at recreational freshwater for our environmental samples. And so the standards they set for E. coli are 126 colony forming units per 100 milliliters of water, and for Enterococci, 30. So to start discussing the methods for our sampling and processing of environmental samples, the first step is the sampling. And so along with our partner organization, Blue Water Baltimore, we sample a variety of locations in the Baltimore region, including Gwynns Falls and Jones Falls, which are two waterways in Baltimore. We have 4 to 6 sampling sites, specific sampling sites at each of these waterways that we return to every time we take these samples, just so we're able to compare levels of our analysis at each sampling trip to the same sites. Then we transfer these water samples to the lab in the cooler within 24 hours, but typically immediately after we collect them. And then this part is key because immediately when we receive our water samples, we do the IDEX assay. And so an IDEX tray, you can kind of see it in the background of the slide. It is composed of a variety, a bunch of small wells and a bunch of large wells. The small wells you can kind of see on the right side. And we fill these trays with 1x and 10x dilutions of our water samples. And we also add one of two reagents to the tray. These reagents, color and alert, after incubating with the water for 24 hours and at 35 degrees Celsius, if there is in fact E. coli or Enterococci in the water, it will react with that bacteria. And that after 24 hours, if you put the tray under UV light, as you can see in the image, they will fluoresce. And so we would count the number of trays that fluoresce and plug them into a formula to determine MPN, which is the most probable number of colony-forming units of the bacteria we're looking for. Colony-forming units is the unit that the EPA standards are in. After this, for the next couple of days, we plate these positive samples onto selective agar. McConkey agar selects for E. coli, and Enterococcus agar selects for Enterococcus. And this is just a way of being absolutely positive that the bacteria that we're looking at is in fact E. coli or Enterococcus before we archive them. And here you can see examples of the bacteria on these plates. E. coli on the left, Enterococci on the right. So the results of our analysis, and I decided to compare the presence of fecal indicators and concentration of fecal indicators in these Baltimore waterways to increasing heat and humidity heat index. First figure to look at is looking at the different concentrations of the fecal indicators per sampling trip compared to the E. coli and Enterococci standards set by the EPA. The red line is the standard for Enterococcus or Enterococci, and the yellow line is the standard for E. coli. The blue columns are the E. coli concentrations for the sampling trip, and the purple is the Enterococci concentration for the sampling trip. And so it is, if the purple line is above, exceeds the red, so if the purple column exceeds the red line, that indicates that the Enterococci presence exceeds the EPA standard. If the blue exceeds the orange, same for E. coli. Something that immediately might jump out to you is that many of these exceed the EPA standards. In fact, only one of these doesn't, which was our first sample at Jones Falls in late March. And many of these actually exceed the standards significantly. For example, in late June at Gwynns Falls, there was well over the EPA standard for Enterococcus. And then, as I mentioned, to compare it to the average maximum heat index 14 days prior to sampling, there was definitely in our sampling trips a connection between a positive correlation between Enterococci concentrations and the average maximum heat index 14 days for the 14 days prior to sampling. This was particularly stark for our samples at Jones Falls, less so for Gwynns, but still positive. So the key results summary, some key results from these graphs is that the beach, as I briefly mentioned, all but one of the sampling trips from both sites showed one or more fecal indicator concentration exceeding EPA standards. Majority of the total samples from both sites contained over 10 times the EPA standard for Enterococci. And there was, as I also mentioned, a very positive correlation between Enterococci concentration, average heat index at Jones Falls. So to connect this to public health, there were high fecal indicator concentrations across the board. As you saw, this poses a significant health risk because these waterways are located in densely populated and often neglected areas of Baltimore. Since these fecal indicators are present in large concentrations in these areas, it's also likely that there are pathogens present in these waterways close to so many people. The connection, the correlation between Enterococci concentration and heat index, there's a possible connection between fecal indicator concentration and climate change, since, as we know, heat indexes are rising year by year due to climate change. A limitation of the study is that it does not actually analyze or detect the pathogens and assumes that they're there. As I mentioned, this is the whole premise behind a fecal indicator bacteria, but there are more in-depth analysis would in fact actually look at the pathogens themselves instead of the presence of E. coli and Enterococcus. Acknowledgements. Thank you so much to my PI, Dr. Goldstein, and everyone at the lab this summer. I think a couple of you are in the call right now. For their patience and guiding and training me. I'm looking forward to coming back in the fall. Also want to thank everyone with CONSERVE for this opportunity. Thank you.
Good day, everyone. My name is Adetutu Boluwatife, and my presentation for today is going to be based on bridging research and public impact through animal waste technologies. I'm a recent graduate of environmental management and toxicology from the Federal University of Agriculture, Abeokuta, here in Nigeria. And beyond academics, I'm very passionate about environmental sustainability advocacy. Yes, I believe that resources should be used in such a way that it benefits us, the present generation, without jeopardizing or compromising the ability of future generations to also benefit from it. And for this year's Global FEWture Alliance Summer Internship Program, I interned under Dr. Stevany Lansing as a virtual intern. And sorry, I have a bit of cough. My work was based mostly on animal waste technologies. And what animal waste technologies mean is they are just various methods and processes that we can use to transform animal waste into valuable resources. And some of these technologies include innovative solutions like composting, anaerobic digestions, and some of the benefits of these animal waste technologies include reducing pollution, improving soil health, and making sustainability practical. Because if we can reduce pollution, things like greenhouse gas emissions, we're able to make our environment a safer space, not just for us, the present generations, but even for our children, our grandchildren, and the future generations to come. And that is actually the goal of sustainability. And we have different animal waste technologies, of which Dr. Stevany Lansing has worked on. We have the anaerobic digestion, which is a kind of biological process where we convert organic waste like manure, food waste in the absence of oxygen into clean energy like biogas. And it also has a kind of byproduct, which we call digestates, that can be used as fertilizer to improve the soil health. We also have composting, which involves collecting and transforming organic waste such as grass clippings, leaves, fruit waste, food waste, and we allow it to decay, and this turns into a nutrient-rich soil amendment to help to improve the soil health. We also have thermochemical processing, which are just processes that we use to turn waste into useful products like biofuel. So it helps us reduce our reliance on fossil fuels that emit so much greenhouse gas into the atmosphere. And also we have biochar. We can get biochar from thermochemical processing. It's a kind of byproduct that we get when we burn biomass in the absence of oxygen through processes like gasification, pyrolysis, or combustion. And it has this sponge-like structure that helps to retain nutrients, it helps to retain water, so it doesn't leach, so it helps to improve the soil structure, and it is also able to sequester carbon in the atmosphere, so this helps to in the soil rather, so this helps to reduce the amount of greenhouse gas in the atmosphere. And during this internship, I have gained a lot of experience. I have gained rich experience, one of which was helping to co-write an article for Journal of Extension. And this Journal of Extension, it focused on how the University of Maryland and the Maryland Department of Agriculture work together with farmers to see how animal waste can be managed on farms in Maryland. Because one thing about improper waste management is that it is not only going to affect our soil, it's also going to affect our air, it's going to affect our water. In fact, it's going to affect the environment at large. And so this is why we really need to be very practical and be very intentional about proper waste management regarding animal waste. And some of the things that I helped to do, I helped in structuring the article, I helped in reviewing and editing the drafts, and also making addition to it. I also helped in making sure that the reference listing and the citation were actually very accurate and correct. And it really helped me in the place of scientific communication, academic writing, teamwork. And I believe that these skills are also going to be very useful for me in graduate school and beyond. Another task that I did during this internship was helping to support research materials and article documents. For the Animal Waste Technologies booklet for University of Maryland, I was able to write the forward section where I wrote simplified and precise descriptions of each of the animal waste technologies, so that somebody that is about to read the animal waste technologies booklet, reading the forward alone, they have an idea of what the booklet is going to be about. And I also helped in reviewing farmer-focused documents, mostly the question and answers, just to make sure that it was very clear and very understandable for the farmers that these documents were to go out to, and to also remove any kind of typographical errors. I also helped in making sure that citations, reference listing for different articles that were going out from the Bioenergy Lab were actually very correct. And this was one of the exciting tasks that I did during this internship. I helped in translating the biochar graphical fact sheet into multiple languages. The biochar graphical fact sheet, it was originally created by Sri Krishna of Sanskriti fans in Nepal, is also a Global FEWture Alliance team. So what the biochar graphical fact sheet is about is just to provide like a visual illustration of how to make biochar and how to apply it to soil. So it's more of like a do-it-yourself method for farmers that is very easy and practical. So if somebody sees this, or they have like a layman knowledge of how they can use resources on their farm to turn it into biochar and also apply it into soil. So the biochar was originally in English and Nepal, but since the University of Maryland's Animal Waste Technology Extension website is mostly translated in English, Creole, and Spanish, so I helped in translating the biochar graphical fact sheets into Spanish and Creole. So here's the Creole version of the biochar graphical fact sheet, and I would say this is like the most exciting. This is the one I enjoyed the most. I helped in reviewing and improving the University of Maryland's Animal Waste Technologies website. So before I reviewed it, this was what the website looked like. It was looking all cluttered with the description of each technologies are just so many long paragraphs with no break. Then for the sections, they had no header. And this is not user-friendly, especially now that we live in a world where people's attention span it keeps declining. So what I did was I took those long paragraphs, I broke it down into clear bullet points, highlighting what the technology is about, how it works, and why it matters for farmers and owners. So, somebody comes into the website the first time and they see this, they are able to like glance through it in like just a few seconds, and they have an idea of what the technology is about already. Then for the sections, I put clear labels. For the fact sheet, I put a recommended for first-time visitors. So, somebody comes into the website for the first time and they see, oh, recommended for first-time visitors. Oh, they are eager to know what is in the fact sheet. Or they say, oh, you have something for me. So they are eager to read what is in the fact sheet. Then under the language versions, I also put a language version. So when somebody comes into the website, they can see that, oh, under this section, we have the fact sheet in different versions. I can either read it in Creole, I can either read it in Spanish. So if you look at what it was looking like before, it was looking all cluttered, not so user-friendly, and this is what it looks like now. So you can see that it's a bit more organized. So another one I did during this internship was helping to simplify complex research on biochar into a fact sheet. So I just looked through different articles on biochar, and I came up with a fact sheet, highlighting what biochar is about, how it works, how it is made, when should you use biochar, when you should not use biochar. It just gives like a layman explanation of what biochar is about. So whether we have a farmer or we have a student or we have a policymaker that is trying to learn what biochar is about in just a short period of time, this fact sheet comes highly recommended. And I feel like that was one of the things I achieved during this internship. It's all about simplifying complex scientific ideas, into something that is simple, that, everybody can relate to. It doesn't have to be, issues surrounding environmental challenges does not have to be anything complicated. It should be something that people should be able to relate to in order to, make better actions to make sure the environment is a safe place to stay. So this internship has shaped my purpose in so many ways. It has strengthened my passion for environmental communication, which is one thing, something I've been doing before. I usually use my social media platforms to talk about issues surrounding environmental management challenges in simple ways, because I'm a sucker for, saying things, in a way that even a child or the unlettered would be able to relate. And it has also helped me in growing my skill in making research relatable and practical through the fact sheets making. And it has also inspired me to pursue a master's in any environmental sustainability related field to, deepen my impact through research and outreach, because I just don't want it to end. And I'm currently looking out for any program in this field, giving out assistantship support, because I want to gain more knowledge. I want to continue working on projects that is talking about how we can address environmental issues through research, through outreach, and through policy making. And I am going to say a vote of thanks to these people, to the Global FEWture Alliance team for this opportunity. I'd like to say a big thank you to Dr. Stevany Lansing. She did not just mentor me, she also guided and empowered me in a very calm and brilliant manner. I would also like to say thank you to Rafian. I'd like to say thank you to Dr. Lina Milai and other Global FEWture Alliance mentors, for investing in my growth during the first week of this internship through the professional development webinar. And, this is just the beginning, and I'm ready to keep learning, sharing, and building impact. Thank you so much for listening.
So, hi everyone, my name is Christien Stevany, and I'm going to talk about the impacts of pretreatments on marine organic matter for dark fermentation. And so here's the outline, or the things that I'm going to talk about. A little bit about myself. I'm studying environmental science at the University of Arizona. I'm also an international student from Indonesia. My research interest is natural resources management, and my personal interest or my hobby is to go on a hike. And you can see on the right is a picture of me went on a hike in Arizona. So this summer, I had the opportunity to work in the Bioenergy and Biotechnology Lab, led by Dr. Stevany Lansing at the University of Maryland. And so this lab mainly works on anaerobic digestion, where it takes waste source to produce biogas, fertilizers, and other valuable products. So I mentioned about anaerobic digestion. So anaerobic digestion is a process that mimics what is going on in our stomach. When we eat food, we produce acids in our stomach with a condition with no oxygen. And then later on, it will be digested as gas. The same thing applies to anaerobic digestion. It consumes organic material. It will later produce volatile fatty acids, and then eventually it will produce biogas. My work only focus on the acid production stage, or this whole process is called dark fermentation. So I only focus on the production of volatile fatty acids here, or we call it VFAs. And so how do I know how much the amount of VFAs are present in the sample? I look at the soluble chemical oxygen demand. So to better understand, you can see the figure on the right. The bigger circles is the SCOD. And within that circle, there are smaller circles, and those are the organic compounds. And one of those organic compounds is the VFAs. And so if there is an increase in the overall SCOD values, it might also indicate that there is an increase in the VFAs concentration as well. So why do I need to know about VFAs or know how much the amount of VFAs in a sample? It is because my lab works in a project called Pod Power, and Pod Power is a bio-inspired battery under the water in the ocean. And there are three main systems of Pod Power. The first one is organic matter collector right here. So it collects organic matter in the ocean, such as phytoplankton, zooplankton, and then after that, the organic matter will go to the fermentation system. This fermentation system is where my lab is mainly focused on. So we try to convert the organic matter into VFAs. And then another group that works on the other system will take the VFAs and convert them to electricity using microbial fuel cell. And so these are the three main experiments that I've been doing this summer. Overall, all of them have similar goals, which is to see how different treatments can affect the amount of SCOD in a sample. And so the organic matter that I'm using for the experiments is spirulina powder. And you can see the picture on the bottom right. This is spirulina powder that I'm using in different experiments. So the first experiment is inoculum and substrate ratio. So I wanted to observe and determine which is the best ratio of inoculum and substrate that can produce the highest SCOD and VFAs. So for this experiment, I started by calculated the sample amounts and load them into the reactor bottles. So you can see from the picture right here, different bottles have different ratio on them, and therefore they also have different amount of substrate in them. And then after that, these bottles went through the batch hydrogen process, where it is a type of dark fermentation process. So these mortars that on top of the bottles has like a stirrer rod, and it will go into the bottles to stir the sample in the bottles. And it helps to break down the organic matter in the bottles. And this whole process takes about 10 days. And so after that, I analyzed the SCOD before 10 days and after 10 days. And then here is the results of the SCOD. As you can see from the graphs, that the ratio that produced highest amount of SCOD is the ratio 1 to 4.5. And on the pH trends, the same ratio, 1 to 4.5, is also one of the ratios with the smallest pH drop. And therefore, the conclusion for this experiment is that 1 to 4.5 is the best ratio to produce the highest amount of SCOD and VFAs. The next experiment is mixture pretreatment. So as I mentioned earlier, that I used spirulina as the main substrate. And spirulina is a substrate that is hard to break down. And therefore, in this experiment, we subjected spirulina with different mixture, such as alkaline mixture and semi-acidic mixture, to see if this mixture can help break down the organic matter in spirulina and will eventually help to increase the SCOD amount at the end. So there are 4 different mixtures that I prepared. You can see here on the table on the top right. And then I measured their initial SCOD and also their final SCOD after 24 hours. And the results for the mixture pretreatment experiment is that you can see that when spirulina is being mixed with KOH, fermentation broth, or the combination of them, they produce higher amount of SCOD compared to just spirulina with water, even before 24 hours. And also after 24 hours, the same thing applies. There are higher amount of SCOD as well. And the third experiment is mechanical treatment, where I pour the water, the spirulina and water into the machine and just hope for the best to see if there is an increase in the organic matter. And so here is the result. The blank samples, the ones that did not go through the machine, have lower amount of SCOD compared to the ones that went through the machine 10 times to around 10 times and 20 times. And therefore, this mechanical pretreatment is also effective in breaking down the organic matter in spirulina. And also it will help to increase SCOD values and also VFAs. So those are the results from my experiments that I've been doing this summer. And overall, what I've gained from the summer experience is that lab experience, starting from collecting samples. I've also got to learn about anaerobic digestion in real-world scale. In the lab, I also learned how to improve my problem-solving skills. I got to work with other scientists and make friends in the lab. I also want to acknowledge Dr. Lansing as my mentor, and also Dr. Hakula for helping me with my experiments, and all my other friends in the lab that has been supporting me. And so, thank you.
So, just a little bit about myself. My name is Alyssa. I'm a rising sophomore undergraduate public health science major. And this summer, I had the opportunity to work with Dr. Davis and Wei, a grad student in Dr. Davis's lab, learning about the effects of field cured products on water quality. We specifically looked at cured in place pipes on the toxicity and electric conductivity of our water. So just a little bit of background information. Cured in place pipes, or CIPP, is a technique that uses epoxy resin to seal or line the inside of our stormwater pipes to protect them from eroding and the elements, etc. And we often see that as these cured in place pipes pipes are lined, or if they're not lined properly, or if the tubes are not lined properly, then we're going to see some leaching of our cured and plate pipe resins into our waterways. We have two examples here. The one on the right is from Virginia, and the one on the left is from Syracuse. And we have found that they are leaching particulate into our water. Yeah. So our study looks at resins that are similar to CIPP resins, but not found in CIPP resins, to understand how these resins would react under similar conditions. And we quantified the toxicity, especially as a result of insufficient curing time. There are other reasons that these cured in place resins may not be fully cured. For example, if condensation and others insufficient curing time. So we do this to encourage improvement in our stormwater pipe repair methods. So our methods are broken up into two main sections: our leachate testing and our acute toxicity testing. So with our leachate testing, we look at three different resins: our steel resin, garage paint resin, and toy resin. And all these resins are similar viscosities and properties of our CIPP resins. And we place them on our glass slides over here. And two glass slides are placed in each vial of 100 milliliters of DI water. And well, first the resin is cured for a few days, different days depending on the type of resin. So 1 day for our steel resin, 1, 3, and 7 days for our garage paint resin, and 1, 2, and 4 days for our toy resin. So after they're cured, we're placing them in our tube, and we're stimulating leaching by putting it in this rotator, which you can see here. Oops, sorry. See here, and they rotate for 24 hours. So before and after this test, we measure pH and electroconductivity of our samples. And then this water that has the leachate in it, that has the particulate from our resin in it, is used as 100% concentration in our toxicity tests, which I'll talk about now. So this is our acute toxicity test. That leachate that I just mentioned is going to be diluted into different increments here. We have 6.25, 12.5, 25, 50, and 100% leachate. And we use Daphnia magna, which are also known as water fleas, as our test organism. They're pre-fed for 2 hours and are placed 5 in each replica of concentration. And they're also placed in a synthetic stormwater mixture with calcium chloride, magnesium sulfate, sodium bicarbonate, and calcium chloride, and stored at room temperature for 6 hours of light and 18 hours of darkness in our incubator. So looking at our results now, sorry, we can see that so this is our electroconductivity measurements. Electroconductivity is basically ionic particles dissolved in water. So when we're seeing more of these higher levels of ionic particles, we know that there's more leached particulate in our water from our resins. We can see we have the toy resin on our left, and we can evidently see that with curing time, as curing time increases, our electroconductivity levels decrease, and that our garage paint resin has a much higher reading. From the start, we have different scales here, much higher reading overall than our toy resin does. So, okay. And this is our pH levels. We can see our garage paint and our toy resin again. We can see that with our garage paint resin, as the curing time increases, our our pH increases in basicity. So, but with our toy resin, we don't see any results in comparison to our garage paint resin and in comparison to itself. It's not consistent with either. And I didn't mention it before, but our control, as mentioned here, is our glass slide with that should say, yeah, is our DI water without resin. So just a glass slide in our water with stimulated leaching and before and after. These are the results for our toxicity test. We can see I measured percent mortality of Daphnia magna with the concentration of our garage paint resin leachate. So we can see that with incremented concentration of our garage paint resin leachate, there's going to be a higher mortality of Daphnia magna, which you could probably have predicted. But our LC50, our lethal concentration to kill 50% of our population, is around 10.5% of the concentration to kill 50% of our Daphnia magna. And here I have a different graph for our, still to measure lethal concentration. So our lethal concentration here is about 7.8%. So that means it's more toxic because a lower concentration can kill the same amount of Daphnia magna. And for our toy resin, we found no demonstrated toxicity. So 0 of our water fleas died within 24 hours of placement in all concentrations. So we don't have an LC50, and we can determine from this that there is no definite LC50 and no toxicity. So some general conclusions from this is that our higher electroconductivity levels are going to indicate more leach ionic particles. We found this more from our garage paint resin and our steel resin than from our toy resin. And we found drastic differences across the different curing times is going to significate the importance of a proper curing time as we go on. And our toxicity test indicates that there's also more toxicants from our industrial resin than from our toy resin. So we hope to use this data to change the way we install and manufacture our cured in place pipes in a way that reduces toxicants in our environment and improve our waterways. I just wanted to take a moment to acknowledge that this is a part of Wei, who I mentioned earlier, his PhD dissertation, and it was nationally funded by the Cooperative Highway Research Program. I would also like to take a minute to thank Wei and Dr. Davis for their support and for everyone at the CONSERVE, Dawn, and Global Future Alliance internship this summer. So thank you.
So our presentation is going with the flow, our experience as CONSERVE interns. I'm Katerina Banovic.
And I'm Alexia Lazo.
Our mentors for this project were Dr. Rihanna, Dr. Lena, and Dr. Suhana.
And so a general overview about our presentation. We're first going to be going over the Maryland Safe Drinking Water Study, and then the Consumer Perception Study, and then lastly, the Anacostia Rivershed Study.
So for our combined efforts, we worked on the Maryland Safe Drinking Water Study, and with this, we assisted in the second phase of the project, and we tested for 4 different kinds of pollutants after collecting water samples from participants' homes. We tested for pharmaceuticals and personal care products, heavy metals, perfluoroalkyl and polyfluoroalkyl substances, which are found in nonstick pans and things like that, and we also tested for bacteria.
And so within our sampling trips, we were able to reach many Western and Eastern Shore counties in Maryland. The collected participants, we collected in total of 9 different counties this summer. We focused more on rural areas, and it was interesting to hear what concerns people had about their water, how they heard about the study, and why they wanted to participate in it.
Okay, so we did some testing within our lab and we tested the microbial samples within our labs. They were processed in the geolabs the day of collection. Samples went through different membrane filtrations and were placed in 3 different tubes. They either went into the non-BRDU non-enriched, non-BRDU enriched, or BRDU non-enriched. And Alexia will explain what BRDU is.
Yeah, so BRDU, it's used as a marker for cell proliferation. It has a similar structure to thymine, and it serves as a building block for DNA. We use BRDU within our testing, specifically during membrane filtration to distinguish between live bacteria and bacteria that may be already dead. And then with the enrichment factor, we use these samples to allow any bacteria in the water to fully grow.
Okay, and the other part that I worked on was a consumer perception study. And for a brief overview of it, it was done because the strain that we put on our natural resources has led us to have to shift towards more environmentally conscious sources of water for non-potable uses like irrigation of crops. And reclaimed water is one of these, reclaimed water being treated wastewater that is then used for, again, non-potable uses. A 46-item questionnaire was developed in Qualtrics and distributed through 2 different formats, either in Maryland Day in 2022 or 2023, and sent out via email to different environmentally focused groups. We received 615 total responses, and only 449 of them were complete, which would be used in further data analysis. Responses were recorded from all over the US, excluding Hawaii, Missouri, South Dakota, Indiana, Kentucky, and New Mexico. And the data had to be cleaned manually because there were some fill-in-the-blank responses that had to be changed. Data was analyzed with chi-squared independence tests and Kruskal-Wallis and Dunn's tests for difference between groups in R. For some of the results and the discussion about what I did, experience with drought and water contamination was linked to region that people were from, as you can see in questions 23, 24, 25, and 26, which are the top 4 graphs. Yeah. And questions 45 and 46 were both linked to people having a higher likelihood of being comfortable with feeding produce that had been irrigated with irrigation water to their family. And then I did a Maryland specific analysis, which was questions 15 and 38. There was significance in looking at people felt that the food that they wanted to eat was available and affordable to them. And most people felt that that they wanted irrigation water to be subject to stricter testing than conventional water. With this, I propose looking farther into what information around reclaimed water is available in Maryland and seeing why that more people feel that it should be subject to stricter testing than conventional water. And I also presented at this student undergraduate research conference, and it helped me gain a broader perspective of my project and the other fields that were there that people presented on. And I got to engage in conversations about my topic and other people's topics. And it was nice to see that they were equally passionate about the science field as I am. And it strengthened my presentation skills, and it helped me find gaps in my research for the manuscript that I've been working on for the consumer perception survey. So I got to do some more data analysis after I talked with people about that. And that's all my information on that.
All right, now I will be talking about the Anacostia River study. So the Anacostia River flows from Washington, D.C. to 2 counties in Maryland. Those are Prince George's County and Montgomery County. And so during the summer, I assisted in processing samples collected from 9 to 10 different areas of the river. And during these testing, we were looking for fecal bacteria such as E. coli and other types of bacteria such as Vibrio and Cephalococcus aureus. And basically with sample processing, we had to go through 3 different procedures or steps. First was the membrane filtration, which is very similar with the Maryland Safe Drinking Water study. But for this study, we used PCB as a dilution to help view growth for bacteria. And we were looking for the 3 bacteria, as I said in the previous slide, E. coli, Vibrio, and Staphylococcus aureus. And second procedure was the bacteria cultivation and preservation. I was able to learn how to streak bacteria on media plates, and this was used for future DNA extraction and PCR PCR confirmation. And the third step was DNA isolation and PCR confirmation. It was used basically to detect for bacteria, to make sure we had a positive indication that it was these 3 types of bacteria that we were looking for. And we also did human nasal and dermal swab tests. We conducted this in Bladensburg Waterfront Park. Participants filled out a survey and consent form. The survey was basically asking what concerns they had about the water, or how often do they use this river for recreational purposes. Overall, we received 30 participants that wanted to join the study. Most of them were regular river users. They used the river for fishing, rowing, and were honestly on the river for at least once a week. And so the samples were streaked for SREs and MRSA, and they were placed on Baird Parker plates and MRSA Select Media plates. And lastly, for our results and conclusion, on the left is the PCR table, and the PCR confirmation showed that most sample sites showed a positive confirmation for E. coli. And on the right with the nasal and dermal samples, and the graph at the top, the BP and RPF plates, about 90% of nasal and 56.66% of dermal samples showed black colonies. And on the graph in the bottom, it basically depicts that on the MRSA select plates, 6.66% of nasal samples showed hot pink colonies, while none, basically 0% of dermal samples showed hot pink colonies. And from this data and collection, we found 10 potential SREs and MRSA colonies picked up from each sample. And so we stored it in TSB glycerol for future PCR confirmations.
All right, and then that concludes our presentation. Thank you guys so much for listening.
Thank you, Katerina and Alexia. I would like to thank all of you all for this wonderful presentation. It's been a wonderful 10 weeks, and I hope that most of you all will continue with us, or will take whatever lessons that you've learned from this internship moving forward.